recording here. And so we were talking about questions on the class. Other questions? Okay. So we were talking about photosynthesis before, and if you remember from where we ended up last time, we'd done this kind of complicated chart for, it's kind of this Z reaction thing that that what I wanted to do is, the first one I wanted to show that we were doing oxidation reduction reactions at both steps. So I was drawing the arrows like we see when we do the electron transport chain, or the curved arrows there. And so what I'm going to do now is I want to redraw this and do the less complicated diagram that you generally see for these sorts of things. So what we have is, we've got water down here, right? So we start with water inputting its electrons to P680, right? P680 is then excited by a wavelength of light that's 680 nanometers to make our excited P680. So in chemistry, we do the asterisk for excited. Those electrons then get transferred down to plastoquinone, right? Which picks up the electrons, which then is going to transfer those electrons to our cytochrome B6F complex. And as the electrons go through, we're going to pump some protons across the membrane. We are pumping those protons into the lumen, or the thylakoid lumen. Then we're going to get to plastocyanin. Okay, then down to P700. Okay, P is protein. Okay, so in this case, sometimes when you see these protein complexes like P53 is, I think P53 is probably, I'm used to these things being named by how big they are, but it's their sedimentation coefficient, at least until something else gets that number. So P53 is kind of how big it is. I'm assuming it's about 53 kD. Okay, heard of P53? Mm-hmm. It's involved in cancer. It's a cell cycle stop protein that gets deactivated. But in these cases, P, it's a protein that absorbs light at 280. That's how they named it. So P700, pretty low, pretty stable in its oxidized or its reduced state right there. And then it's going to absorb light of 700 nanometers. And remember, we've got our antenna complexes, right, that have all the different things that can absorb various energy of sunlight. And they're going to transfer their energy down, bleeding off some of it until you get to something that has this much energy or that much energy. Okay, so we can excite these complexes. That's going to excite up to, so that gives us our P700 excited, which can give those electrons to ferredoxin. Which will give the electrons to NADP+. Okay, it's the same diagram as before, except I'm not showing the reduced and oxidized forms of everything along with those arrows. And so it's less complicated. It's less messy, at least. All right, and then remember that we can have cyclic electron flow. So this produces a certain ratio of ATP to NADPH, 3:2. And then what we've got is this could be cyclic electron flow. All right, and I should actually draw this arrow going to here, because what we're showing is that the electrons these electrons, instead of going into NADPH, can go around in this circle. Questions?
Wait, what? You said going around in a circle. Wait.
The electrons are going around in a circle. So here, if we don't do cyclic electron flow, water's here. We're going to grab 2 electrons from a water. All right. This is not a 2-electron process all the way through. Some of these things are 2 electrons, some of them are not. I'll divide this up in a second. But we pull from the water here. Okay. Pop the electrons up, they come down. Pop the electrons up, they go into NADP+. Okay. And so, and that becomes NADPH, no cyclic electron flow. Cyclic electron flow, instead of giving those electrons to NADP plus to become NADPH, those electrons come back around to here, and we can just run them in a circle and make ATP without making NADPH.
How does that make ATP?
We make ATP because that's a great question. We make ATP because we have a membrane here. We are pumping protons across that membrane. And then what you would do is there's an ATPase in that membrane. And we're going to let those protons back through the ATPase, and it's going to synthesize ATP with that. Does that make sense? Questions?
Wait, sorry, you said there's an ATPase by the cytochrome complex.
There's an ATPase in that membrane. Okay. So if we go back, I'm going to go back to my chloroplast picture from previous class. Let's look at that. So what's going on here with the chloroplast? Actually, here's the picture right here. It's going to be hard for me to draw on here because these are so thin, but we're pumping, protons are getting pumped into the thylakoid lumen right in here, and then you let them out. I don't know what these red dots are, but anybody know what the red dots are? Are they ribosomes in a chloroplast?
Chloroplasts have ribosomes.
Okay, great. Ribosomes, I guess it makes sense they would. They're kind of like mitochondria.
Yeah, they were originally cyanide.
They're like mitochondria, but they're green.
I know.
Anyway, but so we're going to let protons out of these through our ATPase, and we're going to make ATP out here in the stroma. That's going to be important when we think about kind of the regulation of this system. Questions? Okay.
Can you explain, so we talked about the cytochrome B6F, I think you said the hydrogen goes through, can we see that picture?
So back on the other one? Okay. So let's do it right here real quick. Okay. So this is very abbreviated, right? There's the whole Z diagram I just drew is in this. Okay. But you can imagine this is the B in the middle of it. We have a cytochrome B6F complex. It's pumping out of the stroma into the thylakoid lumen. And then we let those back into the stroma to make ATP out here. Let's go to the other, we'll go with the other diagram. Okay, so if I think about this, this is going to be the stroma out here. If I think about relative to that yellow line, and this is a thylakoid lumen above, I'm going to maybe not write it because we've already got a lot going on. Is that?
Yeah.
Okay. Yeah.
In what scenario would they do the electron flow over making?
I don't actually know what the specific conditions are. I don't know why. That's a great question. But I don't know why you do one or why you're doing anything other than the dark reactions with this. But you could imagine that, while the light's going, you're not, while it's light, you're going to be doing dark reactions to store stuff. You could imagine that if you have, certainly if you have a lot of light, right? You've got a lot of light going and you've made all the stuff you want to store. You could imagine you might still want to run ATP to just run things, right? To run various enzymes, and you might run out of this, depending on what's going on. But I don't know what the exact conditions are. But we can get that cyclic electron flow. And there are some organisms. So I don't care that you know what photosystem 1 and photosystem 2 are. It may say in the It may say in the study guide that I do care, but I don't care anymore. So I once cared. But over here, this is photosystem 1. And let's see, green. Over here, this is photosystem 2. And I don't care, so I don't remember exactly where the dividing line is, but it's roughly. And it may be actually, maybe I should be pulling my blue dividing line to include this, because photosystem 1 is some organisms just run photosystem 1. That's it. So they get some electrons and they just run them in this circle to make ATP. So there's some bacteria that do that.
What do you need to go back?
What do I mean, or?
No, what do you need? To make it go cyclic? Oh, we're talking about
What's
energy, or I don't know, I'm not energy, you're making energy.
Some things they have to carry those electrons from either that ferredoxin would have to be right next to this, so it could actually just transfer them directly. I don't know if that's what you're asking. Or there'd have to be some electron carrier that would float around the membrane to do that.
Well, that makes sense.
That may not be what you're asking.
I guess what? So you have to be low in ATP to do this, right? To do the cyclic.
It's when you want when you want more ATP than the 3-to-2 ratio that you need for the dark reactions.
Okay.
Claudia.
So if you were to do the cyclic electron flow, every time you're completing a cycle, are you popping out a proton and that proton is making ATP every time?
I've put one proton. I am not.
Well, not necessarily.
It's not going to necessarily be one. This is going to be 8 electrons. We get 3 ATP from 8 electrons. So we could work out if our ATP works. What was it? It was 4 per ATP before. So we're going to have to pump enough protons. If we're going to make it, we're going to make 8 ATP. I don't want to do math. You guys can do the math. I'll give you the number of electrons, and then you can figure out for how many electrons through how many hydrogens that you would need for that. That would make a really mean question on a quiz or a test. I'm not going to do it. But we could figure it out, right? You guys don't look curious enough, and I'm kind of tired today. So it's not a tangent I'm going to take on my own. Other questions? Okay, so let's talk about that in terms of the stoichiometry of this. This is plasticine or plastiquinone down here. We can look at it in a minute. But this process involves, so in some of these parts of this process, the way it is drawn, you've got 2 electron processes, and some of them are 1 electron processes. All right. So clearly, taking the water, I take 2 electrons per water. I have to get 2 electrons to NADPH. NADP make NADPH, right? Okay. This has got to be a 2 electron process here because I'm going to make essentially, our ubiquinone is that way, and it's going to be our plastic. So some of these things are 2 electron processes, so there's some things going on there. But these are 1 electron processes right here. All right. So what we get, if we think about energy in this process, we have 8 photons that are absorbed. So 8, make that bigger. So 8 photons are absorbed. And what I mean by that is I mean 4 680 nanometers and 4 700 nanometers photons. So how many electrons are we moving through this process for what I'm talking about? If I'm talking about 4 680 and 4 700, and I just told you those are 1 electron processes. So we're going to do this 4 times. So it's not going to be 8. Do this 4 times for 4 electrons and this 4 times for those same 4 electrons as they move through. Okay, so 4e minus.
Right, because 4 times 4 is 4. Wait, what?
Are we good?
No,
Okay,
but keep going.
so we have 4. So we pump 4 electrons here. There's I pump an electron here. It goes through here. I pump it up again here, right? I do that 4 times. 4 times each for 4 different electrons.
So there's 4 total electrons that go through the entire chain.
There's 4 total electrons that go through the total thing for the number of ATP and NADPH I'm about to tell you. Does that make sense?
Yeah.
Okay. 2 electrons could go through it to make how many NADPH? If I have 2 electrons go through, how many NADPH do I make?
1.
1, exactly, because NADPH and NADH are 2 electron carriers, right? Right. That's how we can ground ourselves in this confusing thing that's moving back and forth between whether it's a 2 or 1 electron carrier. All right. So for 4 electrons, I'm going to get 2 NADPH, right? Okay. And we can't do this for ATP exactly. We're going to get 3 ATP. Okay. And this is the ratio that I need. We'll see this. These are the ratios we need for the next process, for the dark reactions. Questions before I move on to dark reactions? I'm going to move on to the oxygen evolving complex quickly. Okay, so this is a big deal, taking electrons away from water. That's pretty hard, and we've got a very interesting structure that's involved in that. This is the oxygen evolving complex. All right, so the oxygen evolving complex is going to pull electrons away from water. All right, we're going to have 2 waters here to get 4 electrons. We're going to get those electrons one at a time, not necessarily from the water, but they get transferred to P680 one at a time. So P680 picks up an electron, it gets excited, it gives the electron away, right? That goes down the Z diagram. It then picks up another electron, it gets excited, it gives that electron over. So we're going to do that 4 times across this complex. This is an interesting complex, and it has manganese in it. Okay, manganese, very oxidizing metal. And so the thing I think is interesting about this, or one thing I think is interesting, is this is one of those examples. Remember, for vitamin E could form a very stable radical. So for vitamin E, it's an antioxidant because it can have an unpaired electron and it's okay. Tyrosine's the same way. So the way tyrosine is going to do this process, we can have a tyrosine that forms a radical. In other words, you could take that electron away from it, and then we can give it another one. Questions? It's called the oxygen evolving complex because it's why we have oxygen in our atmosphere. Does this figure, I'm not necessarily going to ask you to describe this figure, but does this figure make sense in what we just talked about? We had 4 electrons that were going to go through our light reactions, right? We had four excitations of P680 to move those 4 electrons along.
Where's the other 4 like protons associated?
I don't know.
Okay.
They just go somewhere. It's like, okay, questions?
So, tyrosine.
Tyrosine could form, what I really think is cool about tyrosine is it can make these stable radicals. And so we see that here where it's useful, right? What you've got is, you've got P680 oxidized, right? P680 oxidized is this for your tyrosine. It picks up an electron. If you excite it, it gives up that electron.
Oh, okay.
And it comes back to looking like it's oxidized like this. And if you remember, you probably don't remember the structure of vitamin E, but vitamin E has, I think it has a hydroxyl group off of an aromatic ring, so it can do the same sort of thing. That's how it acts as an antioxidant. Okay, the dark reactions. So the dark reactions of photosynthesis occur in the stroma. Can you guys see that in the back, or should I make it bigger? What? You can see it, Jacob? Okay. Okay, they're called the dark reactions because they don't use light. All right, so somebody in here called them the light independent reactions is maybe what they're saying in biology, which is more correct. Okay, apparently hasn't made it to the biochemistry textbooks. Okay, so what was that? It does sound cooler. I agree. So dark reactions of photosynthesis, they occur in the stroma. They are called the dark reactions because they don't use light. Okay, they do not occur in the dark. Because the light reactions aren't making the ADP and the NADPH they need in the dark. Does that make sense? Okay. Questions? All right, so we've got a kind of, it's not that complicated diagram. This is also called the Calvin cycle. Okay, that's the Calvin cycle. And essentially what is going on here, I'm going to summarize it. You can see it actually happening here, but overall what's going on is we start right here, okay? The number of each one is right here, okay? We start with 3 times 5 carbon molecules, all right? We take 3 carbon dioxide and we add 1 to each, and we end up with 3 times 6 carbon molecules, okay? Those break down to make 6 times 3 carbon molecules. It's got 3 6 carbon molecules, they split in half, all split in half, we now have 6 3 carbon molecules, right? And then we regenerate these 3 5 carbon molecules and we take 1 times 3 carbon molecule off. So for each cycle, and that's shown right here, we're going to take, in fact, that molecule is glyceraldehyde 3-phosphate. Remember that from glycolysis? Right, it's right in the middle? Yes.
Where I'm counting 3 carbons for 3-phosphoglycerine, where are the other?
You're counting 3 carbons for
You said it was, is it?
for 3-phosphoglycerate. There's 6 3 There's 6 3 carbons of those.
So where's the 3 times 6 carbon?
It's in here.
Okay.
So they blended that step together there.
Okay.
Okay. So these, if you think about it, this very first step is this. Okay. And that first step is catalyzed by an enzyme called rubisco. Okay. That's the enzyme. I'll just say it's an enzyme that catalyzes that. And it's an important enzyme. Questions?
So why is it 6 carbons? Is it because 3 CO2s enter and then?
3 CO2s enter. So you got 3 5-carbon molecules, and we start there.
Okay.
It's a cycle, so there's exactly start right. We start there, 3 5 carbons. We add 1 carbon to each of them.
Okay.
Let's look at, before we move on here, let's go ahead and look at the number of ATP we use. Let's count them up. I've got 6 here, right? Okay. And I've got 3 here. So that's 9 ATP used, right, to do this. And then, so we'll talk about energy used, energy cofactors used. Okay, we've got 9 ATP, and we've got how many NADPH do you see being used? 6 NADPH. Okay, so our ratio, we think about our ratio there, we've got 6 NADPH, we've got 9 ATP. What was our ratio of these coming off the light reactions? How many did we make when we did one of those runs for the light reactions? 2 and 3, right? So 2 NADPH and 3, oh, I'm sorry. Yeah, no, 3 ATP. See, those are the same?
Mm-hmm.
So we're getting these things off of the light reactions, ATP and NADPH, in the ratios we need for the dark reactions. All right. So you've expended this much energy to make 1 glyceraldehyde 3-phosphate. That pops out here. All right. So it says energy production via glycolysis. Okay, somewhere, but really we're trying to make sugars or starches to store here. Right. You could take the glyceraldehyde phosphate and use it somewhere directly to make ATP in another part, but in the chloroplast, at least, you're already making the ATP directly with the light reactions while this is going on. Questions on where we are? Okay. I want to talk about, well, actually, let's quick talk about or remind you what this glyceraldehyde 3-phosphate is good for. So here it is, right here. Okay. So when they talk about energy usage, you could take that glyceraldehyde 3-phosphate into glycolysis and run it down this way. You had to ship it off to some other part of the plant, right? ATP is not something that really gets shipped between cells when you think about it. So you don't have ATP in your blood. If you have ATP in your blood, it means that something's ruptured, and it's like one of the things they might, I don't know if they test for ATP directly if you have a heart attack, but there's definitely a whole series, there are enzymes that they test for that come out of ruptured cells to test for heart attacks and things like that. But ATP shouldn't be in your blood, period. So you might ship this is plants, but they're not going to ship ATP independently. They might ship this around directly for immediate needs while it's light out. Otherwise, they're going to store it. And so we could store it by running it up gluconeogenesis to make glucose, and then you could take that glucose and make it into starch. Right? And you should be comfortable with that. Okay, after we've talked about those. Questions? All right, then what I want to do is talk more about this Rubisco enzyme. All right, so it's catalyzing this first step. It's doing a lot of stuff here, right? It's running us through this for most of the first part of the cycle. There's a lot of stuff going on in the regeneration of those 5-carbon intermediates as well, as you'll see. But that's super complicated, and I'm just going to show it to you to show you that it's super complicated. All right, this talks about the oxygenase activity of Rubisco, but I don't want to talk about that just yet. Okay, Rubisco, here's some characteristics of it as an enzyme. It's slow. Okay, so its kcat is equal to 3 carbon dioxide, so it's fully saturated per second, which is a pretty slow enzyme when you think about it. Actually, MTHFS that you're working with in lab is also a pretty slow enzyme. Its kcat is about 1 per second. Let me go ahead and get this fixed. All right, so it's a slow enzyme, but what it's doing, this is responsible for like all life, right? All life, this is responsible for capturing energy for the sun and making the rest of life possible. So you have to have a lot of it. It's like 3% of the dry weight of a leaf, of leaves. So 3% of that dry weight is one enzyme, Rubisco. It's the most abundant enzyme on the planet. It's also the most abundant protein on the planet. Proteins do structural things as well. Okay, it's not very specific when we think about enzymes. We think about enzymes having one substrate, right? Well, it can actually use carbon dioxide or oxygen as a substrate. I'm just going to write it out so I don't have to do the subscripts. All right, so not very specific in terms of that. And it has 8 active sites when you think about the enzyme complex. But it's not but they're not cooperative. What would it mean? What does it mean to say that they're cooperative if they work cooperative?
They work with other enzymes and stuff.
They don't work with other enzymes, they work with each other, respond to each other. So hemoglobin is cooperative in that when an oxygen binds to one of the sites on hemoglobin, the other ones get better at binding oxygen. Okay? Enzymes do the same thing. They're enzymes that if they have multiple active sites, that when one is active, the other ones become more active. Okay? But this has 8 active sites and they're all independent of each other, so they don't take advantage of that sort of thing. Questions? Okay, so we got this big, slow enzyme. And not only is it slow, but it actually will use oxygen sometimes. So here's an example. So the carbon dioxide activity of it is this reaction, right? The carbon dioxide activity is taking ribulose 1,5-bisphosphate and taking it down to 3-phosphoglycerol. Okay? The oxygenase activity, instead it takes an oxygen. Once again, we have our ribulose 1,5-bisphosphate right here. It's going to react that with oxygen, and we break down, we get, instead of getting the phosphoglycerates, right, 2 phosphoglycerates for one of those, we get a phosphoglycerate and, make this a little bigger. And we get this 2 phosphoglycerate off of it. All right, so what we've done here is we made one of the things we normally make by adding oxygen, but we've also got this 2-phosphoglycolate off of it. And that's inefficient. It requires salvage. Okay, it's going to take energy to regenerate that.
Like in general, it requires that, or?
Oops, sorry. Let me finish writing here. So it always does. Whenever you have an oxygen react, you have this happen. All right, so we've got this enzyme. It's been around forever, right? It's been around since oxygen, since the air had oxygenated on Earth. So we've got billions of years here. So why is that? So why does the oxygenase activity exist? It must actually exist for a good reason. There's been plenty of time for something to evolve that's more efficient than this. All right, so there are a couple thoughts on this that I know of. I don't know what have your biology professors talked about this at all?
Probably.
Okay. If you can give me what you think their favorite, they may have looked at this or thought about this more than I have. So one thought is that Rubisco evolved before there was O2 around.
Oh, yeah, that's what.
And sure, I guess, I don't know I like that one as much. It doesn't mean I'm right, okay? It seems to me that something could have evolved. It's had oxygen, there's been oxygen around for a long time to deal with that. There are, of course, systems that, so if you think about, we have the same 20 amino acids, right? We'll learn more about this, but you have a codon, and it has to match a tRNA that has an anticodon, and then you have an enzyme that loads the right amino acid on. You get multiple pieces that have to work in concert with the codon and the tRNA and all that. And if you were to have, for that to actually change from the 20 amino acids we have, you'd have to have multiple systems mutate at the same time. Okay, so when I see that we have 20 amino acids and they're pretty much the same everywhere and the same codons are used, I kind of believe that, right? It's kind of like, okay, it's so improbable that you get all these things mutating at the same time. If one of them mutates, the organism just dies. So maybe something's going on like that here. But it seems like it's one enzyme to me. So from my perspective, I'm going to go ahead and put that up there, and they like it in cell bio, and great. They may be right. But what I like better is it's somehow important to bleed off energy. Have you heard that one? So if you're a plant and it's sunny out and you've got all the ATP in it, you've got all the starch you need or can store for now, you're going to have to bleed off energy somehow, right? So if it's really sunny out, you need to deal with it. You can't go in the shade, okay? And so one thought is that this is a way you run this reaction, you use oxygen, and you bleed off energy because you can't really use it anyway.
Isn't doing CO2 like disruptive sort of
Isn't
like Rubisco, like you don't want it to bind to CO2, right? We don't want it to bind to oxygen.
you don't want to bind oxygen. CO2 is the main job it's doing,
Okay.
but it has this oxygenase activity that's been around for billions of years. So I like to think there's a reason for that. Your biology professors have thought a lot more about evolution probably than I have, or maybe at least more. Okay, so we've got that right. Any more on that? Talk about Rubisco there. We'll talk about Rubisco regulation next time. It's really cool the way that works in with photosynthesis. But what I'd like to do for the dark reactions is I'd like to cover one more thing and then let you guys go. So we've got this part here where once we have our glyceraldehyde 3-phosphates, or once we could be here too, in either of these spots, we've got 6 3-carbon intermediates, right? And what we need to do is we need to somehow make them into 3 5-carbons. Okay? So, how does that happen? Well, that's complex. So turning 6 times 3 carbon intermediates, it's really 5 times at this point. 5 times 3 carbon intermediates, because you pull one of them off, right? And you use it. Carbon intermediates into 3 times 5 carbon intermediates is a complex process. And that's really all I want you to get from this figure. But let's go ahead and look at it, and let's identify where everything is real quick, and then we'll be done for the day. Because I know you're all itching, or most of you are itching to go take your micro exam. So let's go ahead, and on here we've got a complex set of enzymatic reactions going here. A bunch of stuff's going on, right? Let's just go ahead and identify our input, right? Our 3 carbon inputs. So what do we see? We need to find 5 of these. So here's a glyceraldehyde 3-phosphate. Here's a dihydroxyacetone, right? 1, 2. Here's a dihydroxyacetone phosphate. Sorry, I should have said it earlier. Glyceraldehyde 3-phosphate. One more. Glyceraldehyde 3-phosphate. I don't think I missed any. There are 5 3-carbon intermediates coming in. You remember that dihydroxyacetone is in equilibrium, right, with the in glycolysis with glyceraldehyde 3-phosphate. And then so we need to make these 5 carbon molecules. So here we have a ribulose 1,5-bisphosphate, and here we actually have 2 of them. It's just not shown. Okay, so they don't put a 2 there at all. Times 2. Okay, but you've got this complex set of reactions with a lot of different enzymes that are going to get us there. Questions? Okay, well, you guys have a great weekend. Has everybody moved their plates? Okay, if you haven't moved your plates, let me know right away so I can open things up for you. Did everybody have colonies on your site-direct immunogenesis plate, or you've told me? Okay, good.
For mine, so I'm just going to use the sample stuff.